I would like to start by extending a warm welcome to all of you and thank you for being with us today. My name is Ann-Sofie Forsberg and I am the product manager at Nidacon Sweden. Today it is my pleasure to introduce our speaker Emma Holmes to you. Emma has worked at Nidacon for the past 15 years and she is today our product development manager. Emma's educational background includes a bachelor degree in science from the University of Guelph in Canada. She graduated with honors with a focus on animal science. After completing her education in Canada, Emma was longing for a return to her native country, Sweden. Once returning to Sweden, Emma's thirst for continued knowledge led her to begin working with her thesis regarding osmolality and sperm function. The work was completed over several years at Karolinska University, Stockholm, Sweden, under the supervision of Professor Ulrik Kvist and Dr. Lars Björndal. And in 2020, Emma received her PhD in medical science. Emma's thesis consists of 4 papers, of which 3 are already published. Her research covers the very important topic of how to treat sperm in the lab. More specifically, a focus on examining the osmolality changes and its consequences for sperm motility and sperm selection in the laboratory. Emma is also a sitting member on the board of directors for the Swedish Society of Andrology. So may I present to you Emma Holmes.
Hello, my name is Emma Holmes. I recently finished my PhD at the Karolinska Institute in collaboration with the ANOVA clinic at the Karolinska Hospital. And I will give a short presentation of my PhD thesis on factors affecting human sperm function in vivo and during processing for assisted reproductive treatments. I will start by giving you some general background on osmolality. The definition of osmolality is the concentration of osmotic particles in a solution. Cells in general are dependent on the osmolality being controlled in their surrounding environment. This is done through channels in the cellular membranes. Particles and water can be transferred between the intracellular environment and the extracellular environment. Sperm are unique since they leave the homeostatically controlled environment in the human body. In order to create and maintain a transmembrane equilibrium of osmotic pressure. That is, the number of particles should be the same inside and outside of the cell. This mechanism of passive and active membrane channels prevents hypoosmotic or hyperosmotic stress in the cell. Such stress can cause damage to the cell, or it can even lead to the cell rupturing or shrinking. The osmolarity of distilled water is 0, since there's no particles present. The osmolality of blood plasma is around 290, which is a physiological osmolality. The osmolality of human urine can be between 50 to 1200 milliosmol, depending on whether the person is dehydrated or has recently drank a large quantity of water. This is depending on the functions of the kidneys to regulate the volume of body fluids and fluid osmolality. All of our pure sperm gradients have an osmolarity of between 300 to 310 milliosmol. Pure sperm buffer also has an osmolarity between 300 and 310 milliosmol. Pure sperm wash, on the other hand, has a lower osmolarity between 290 to 300, and this is because of the gradual decrease in osmolarity that the cells will encounter when going into the culture medium. This osmolarity has been adjusted specifically since it comes in contact with the sperm. The sperm sample most likely starts at 290 at ejaculation. As mentioned previously, the sperm cell is unique since it leaves the homeostatically controlled environment in the body. It also has a unique shape since it needs to be motile. The ejaculate as we know it, collected in a container in the laboratory, only exists there. During ejaculation, the sperm and seminal fluid is expelled in a specific sequence. The first fraction is the sperm-rich fraction, where most of the sperm are expelled. The second fraction is the prostatic fraction, containing secretion from the prostate. This fraction has a relatively high concentration of zinc, and it also contains the enzyme serine protease. After that, there is a gradual increase in fructose concentration in the following fractions. These fractions are also high in peptides and proteins. And this is the secretion from the seminal vesicle. When these fractions are mixed in the laboratory, there is a gel formation followed by an enzymatic breakdown of large protein as the semen sample liquefies. This is the first article in my PhD, and here we wanted to examine what happens to the osmolarity in a semen sample after ejaculation and collection. So we looked at the change in osmolarity in 47 semen samples incubated for 3 hours at 37 degrees. The mean osmolarity after 1 hour was 322 milliosmol, with an average of 280 to 361. The mean change in osmolarity after 3 hours was 57 milliosmol, with a range of 13 to 105 milliosmol. The final osmolarity had a mean of 379 milliosmol, with a range of 292 to 454. As you can see, there is an individual variation between these samples. This is important since we use media such as gradients and swim-up media that most likely is adjusted to a physiological osmolarity of 290 to 300. If the sample sits for 2 hours after ejaculation and increases by possibly 80 to 90 milliosmoles, this means the samples will be exposed to a large decrease in osmolarity, coming down again to 290 to 300 milliosmol. So now we wanted to know how temperature affected the increase in osmolarity. Here we looked at 6 pools of liquefied semen, subdivided into 4 different temperature groups. As seen in the graph, one group was kept at 37 degrees, one at 18 to 22 degrees or room temperature, one group was kept at 4 to 8 degrees in the refrigerator, and the last group we kept in the freezer at minus 20 degrees. We saw that the samples stored at 37 degrees increased significantly more than the samples stored at room temperature. Samples stored at room temperature increased significantly more than samples stored in the refrigerator, and the samples stored in the freezer did not increase at all. We then wanted to see how the change in osmolarity was affected by very high temperatures. This was done since we suspected that the increase was caused by the enzymatic breakdown of large protein into smaller protein, thereby increasing the number of particles. We know that 100 degrees Celsius will stop any enzymatic activity. So here we can see 6 different semen samples that were split in half. One part was exposed to 100 degrees for a short time period, and the other part was kept at 37 degrees. The osmotic increase was completely stopped in all the parts exposed to 100 degrees. We do see a very small increase right at the start, and this is most likely due to the time it takes for the sample to come up to a denaturing temperature for the enzymes. And now we wanted to investigate what factors might influence the change in osmolarity. We looked at the osmolarity in whole semen compared to sperm-free seminal fluid to see if the sperm somehow could affect the increase in osmolarity. The graph on the top left side, on the x-axis you see the different concentration, on the y-axis is the osmolarity. And we can see here that the sperm in the samples do not affect the osmolarity, since the concentration doesn't change the osmolarity. The second graph in the middle shows WS, which stands for whole semen, and SF, which stands for sperm free. You can see that the 5 samples change just as much in osmolarity, whether the sperm are still there or have been removed by centrifugation. In the last graph to the right, we measured the osmolarity in azoospermic samples and samples from vasectomized men and compared to whole semen samples. As you can see, there are some outliers, but the average osmolarity is the same in all 3 groups. We concluded that sperm do not affect the osmolarity. It doesn't make a difference if we remove them or if they've never been there to begin with. We then wanted to see if diluting the samples would affect the increase in osmolarity. 8 different samples were split into 2 aliquots. One aliquot was diluted with Earl's salt solution containing 10% HSA. Osmolarity was measured in the diluted and the undiluted samples, right after dilution and again after 3 hours. The diluted samples only increased by 22 milliosmol compared to undiluted samples, which increased 68 milliosmol. In the diluted samples, there was also a smaller variation between the samples. We were then curious to see if the composition of the ejaculate would affect the osmolarity during 3 hours. So osmolarity was measured in 48 semen samples after liquefaction, and again after 3 hours of incubation at 37 degrees Celsius. The levels of zinc, which is the prostate fluid marker, the level of fructose, which is the seminal vesicular marker, and alpha-glucosidase activity, which is the epididymal activity marker, as well as sperm concentration was recorded. The concentration of zinc was positively related to the final osmolarity after incubation. So the relation between prostatic marker zinc and increase in osmolarity during 3 hours, we could see that 19% of the variation in final osmolarity was correlated with the zinc concentration. So we concluded that the prostatic fluid containing zinc and large amounts of enzymes is what drives the increase in osmolarity. One can guess that the samples that increase the most have large prostatic fluid fractions. So in the 3rd publication, we wanted to look at how this change in osmolarity affects the basic functions of the sperm, in this case the motility. We therefore wanted to expose the sperm to an osmotic challenge, similar to that which can happen in the lab when samples sit for long after ejaculations. 6 different semen samples were washed according to a standard density gradient protocol of 300 milliosmol. The samples were then washed and split into 5 aliquots and mixed with more washing medium of varying osmolalities. As you can see in the table, the final osmolarity of the samples were either 300 milliosmol, 450 milliosmol, 150 milliosmol, or 112 milliosmol. Directly after mixing the samples, we analyzed them by computer-assisted sperm analysis. We looked at the curvilinear velocity, which is the green line seen here. We also looked at the average path velocity, or the VAP, seen here as the red line. Finally, we looked at the straight line velocity, or VSL, seen here in the blue. When the sperm were exposed to a decrease from 300 to 112 milliosmol, all the sperm stopped moving. Motility was equal to 0. When they were exposed to a decrease from 300 to 150, the VCL decreased by 50%. When they were exposed to an increase from 300 to 450 milliosmol, there was no change in VCL. It seems like the sperm are not as sensitive to an increase in osmolarity as they are to a decrease. We then wanted to look at how a change in osmolarity from 400 to 290 milliosmol would affect the sperm. 8 semen samples were washed according to a standard density gradient protocol, this time with an osmolarity of 400 milliosmol. Sperm were then mixed with washing medium with different osmolarities. The final osmolarities were 400 milliosmol, and this was the control, or 290 milliosmol, which was the test. This represents an increase to 400 that might occur in the lab if the sample is left sitting at 37 degrees, after which we add selection media that may have a physiological osmolarity of 290 milliosmol. Motility was analyzed directly after. So here we can see the results. In graph A, we can see the curvilinear velocity. In graph B, we see the average path velocity. In graph C, we see the straight line velocity. In graph D, we see the progressive motility. In graph A, we can see that the test group, that is, the sample going from 400 to 290, curvilinear velocity on average was 20% lower than the samples that were kept at 400 milliosmol. Average path velocity also decreased but not by as much. Straight line velocity did not decrease at all on average. The progressive motility only decreased a small amount. Here we see a short recording of the semen sample that goes from 400 to 400 milliosmol, and this is again the control. We can see the typical beating movement of the sperm tail, or the whipping. And when the film stops, we can also see that the sperm tails look normal. This recording is of a semen sample going from 400 to 290 milliosmol, so this was the test. We did see that something happened to the tails, and that was that they folded or coiled. It almost looked like something was holding onto the tail, and the sperm didn't have the very wide and whipping or beating movement that we normally see. While analyzing the motility of the sperm, we noticed that the sperm tails coiled and folded. We wanted to look closer at this phenomenon. In fact, it has been observed before in animal sperm many years ago. In order to mimic the variation in increase in osmolarity that we see in different samples when they are stored in the lab. So we designed an experiment where we adjusted the osmolarity of the density gradients. As you can see here, we treated the ejaculate either with a 350 milliosmol gradient, or a 330, a 310, or 290. We then treated them with washing medium, also with varying osmolarities, either the same as the gradient that they had been selected with, or we brought them down to 290. Samples were then fixed and analyzed under phase contrast microscopy and judged based on these 4 criteria of tail coiling. The first one being more than or equal to 50% of the tail being folded. The second being less than 50% of the tail folded. The third being the tip of the tail being coiled. And the fourth being a normal tail. So in the samples that were exposed to 330 milliosmol and then brought down to 290, 18% of the tails coiled. In the sample exposed to a gradient of 350 milliosmol and then brought down to 290, 57% of the tails coiled. In the samples where the osmolarity of the gradient was 400 milliosmol and then brought down to 290, 89% of the tails coiled. In the final article of my PhD, we looked at how handling of the human ejaculate affects osmolarity and yield by density gradient centrifugation, and also how DNA integrity is affected. In this study, we stored the semen at 37 degrees in order to get a large increase in osmolarity before selection. So the samples were incubated at 37 degrees, and after 60 minutes, and then again after 180 minutes, the osmolarity was measured prior to layering the samples on a gradient. After the gradient selection, sperm concentration was assessed. We noticed that the yield decreased with increased storage and increased osmolarity. So the yield decreased from 9.5% to 5.3% after 3 hours of storage at 37 degrees. Or from 3.2 million sperm at time 0 to 2 million sperm after 3 hours. This is important since during art, sometimes the yield will be used as an indication to what treatment should be used for the couple, IVF or ICSI, for example. We also looked at how defragmentation index changed after storage time, and we could not see a difference in DFI with different storage times after ejaculation. And finally, the latest update on this subject. An article published by Punjabi et al. In November 2020, he showed that the odds of a pregnancy outcome after IUI was doubled when the time interval between collection and IUI was kept below 21 minutes. So this shows that the time between collection of the semen and preparation is very important, even for the pregnancy outcome. So to summarize, the osmolarity of semen samples increase, and this increase varies greatly between individuals. The increase is likely due to enzymatic factors in the semen. The relative contribution of the prostatic gland can explain 19% of the variation in increase. The increase is temperature dependent. Diluting the samples significantly reduces the increase in osmolarity. The increase will affect the results of selection and preparation for art, both in terms of motility patterns and yield. So what can we do to minimize this effect? Should we use a standardized protocol for all semen samples, or should we individualize protocols? Can we dilute all the samples as soon as possible after collection? Can we minimize the time between collection and processing somehow? Thank you very much for listening, and if you have any questions at all, don't hesitate to contact us at Nidacon. All the contact information can be found on our website.